Welcome back. It happened one night. Age 61, Alan was used to awakening often to urinate at night. He really didn't think much of it, just old man stuff he had been kidded about on his 60th birthday party. But this time was different. The urine was just dribbling out and it looked red. He was alarmed and went to his primary care doctor the next day. Looking at Alan's urine specimen under the microscope, the doctor saw blood cells, and a quick test for bacteria in the urine ruled out the common urinary tract infections. So the doctor asked Alan to get up on the examining table. Doctor put on a sterile glove and did a digital rectal exam in which he inserts a finger in the rectum and felt Alan's prostate. This is a prostate exam. And what he noticed was that the prostate was enlarged. And so the urine flow was being blocked by the enlarged prostate, especially when Alan was reclining. But it could be benign because prostates do get larger as men get older. But as the doctor was feeling around with his finger, he noticed an irregular bump on the prostate gland. And so he sent Alan to a urologist. A urologist is a doctor who specializes in diseases of the urinary system, the kidneys, and the prostate. And the urologist took a biopsy. And in diagnosing prostate cancer, biopsy is very important. And they take 12 core needles. In other words, they take needle, put it into the prostate gland where the tumor is, and he took 12 cores, tiny cores. He sent the samples of this prostate to a biopsy to a pathologist who looks at disease. Pathologist looked through the microscope and found, examining the cells, prostate cancer was present in all 12 cores. 2 weeks later, Alan was treated with surgery to remove the prostate, along with the cancer, entirely. Alan recovered well, and every couple of months he would get a blood test for a marker called PSA, prostate-specific antigen. PSA is a molecule made by normal prostate cells when they're dividing and prostate cancer cells when they're dividing. But, Alan's prostate was gone. So any PSA is going to come from, presumably, from metastatic prostate cancer cells hiding somewhere in the body. But post-surgery, for months at a time, his PSA level was virtually undetectable. And this was really good. No prostate cells anywhere, cancer or normal. However, 2 years later, Alan noticed pain in his lower back. And he mentioned it when he saw the urologist for his PSA checkup. This time, the urologist examined PSA and it was beginning to increase. So the urologist, looking for metastases, did a bone scan for tumor cells on the bone. I described this in an earlier lecture where a radioactive element replaces calcium that is used in the bone. And it's called technetium. And what he found was a picture that we've seen before, which is bony metastases in the back and in the elbow. Now Alan's cancer, which had come back, previously it was in remission, you couldn't see it detected anywhere, it had come back and it's called recurrent cancer. Now this time the doctor tried a new treatment. Obviously surgery is not going to work because you can't, when you've got a metastatic tumor, you can't kind of usually pick out the metastases, like go into the elbow here and just pick this one up, because those are just the bony metastases. There might be metastases elsewhere that are not on bone. So he's going to need a treatment that's essentially chemotherapy, a chemical that will go all over the body and try to kill prostate cancer cells. Prostate cells are stimulated to divide by testosterone, the male sex hormone. So to lower Alan's testosterone production, the doctor used a drug that we are familiar with from the last lecture. It's a drug that blocks the synthesis of testosterone. In this case, the drug blocks the brain region that controls testosterone production. This is called, for want of a better term, chemical castration. The old way, they used to actually use actual castration, removal of the testes. But here we're doing chemical castration so the testes don't get the signal to say, hey, make testosterone. It was successful. Alan's PSA levels went down to nothing, and things were doing really well. The bony metastases were gone. He had no pain. Things are just fine. After 14 months on this regimen of this inhibitor of testosterone, PSA levels started rising again. In other words, the drug wasn't working. In other words, the tumor, recalling from the previous lecture, was resistant to the anti-testosterone treatment. It no longer was testosterone dependent. Now, until recently, the clinical course of a patient with testosterone-resistant cancer was inevitable and dismal, typically 2 years to live. Allen's urologist, however, tried a new personalized approach to harness Allen's immune system to reject the tumor. He tried to make a cell vaccine that was a self-vaccine. A cell vaccine from Allen's cells that was a self-vaccine, Allen's cells. Self-vaccination. To do this, first, white blood cells were removed from Allen's arm. So he just took a venipuncture and removed some of these immune system cells from his arm. The immune system cells were brought to the laboratory and they were stocked. That is, they were S-T-O-C-K-E-D, not stocked as in following. They were stocked with a special protein. They were Allen cells, but in the lab, this protein made by biotechnology was put into those cells on their surface. There were two parts to this protein. First, part of it is an immune stimulant that, if you go back into the body, would say, hey, reject whatever has this particular flag. And second was a surface protein normally present on prostate cells. So that is kind of a double whammy. You got an Allen cell with this thing sticking out, this protein sticking out that says, come kill me, and I'm a prostate cell. So hopefully the immune system will be harnessed. The cells with this added value were expanded in the laboratory over a couple of weeks, and they then were infused back into Allen. So they were put back into his arm. And what this did was stimulate Allen's own immune system to reject not only these cells that had the come hither and reject me, but also prostate marker. So it rejected all the prostate cells anywhere they were in the body. And the only prostate cells at that point in the body are prostate cancer cells. Allen's condition improved somewhat, and he lived longer, although he was not cured. But it's an exciting new approach, harnessing a patient's own cells to reject the tumor. Two issues. Number one, what was the benefit of this vaccine? The survival benefit of this vaccine is typically 4 months on the average. For Allen, it was actually 9 months before his cancer returned, and sadly he died. Second issue, the cost. Doing all of this biology in the laboratory cost Allen $100,000. So let's look at it. His life lasted about a year more, so about $10,000 a month. For most patients, it's 4 months. That's $25,000 a month. Allen's insurance initially said, we're not going to pay it. It's too high a cost. But lobbying by patient groups was very active, so insurance does pay for this vaccination now. These two issues are at the frontiers of cancer treatment. First, effectiveness, and second, cost. In this lecture, I'm going to describe two aspects of cancer treatment at the frontiers. First, the immune system being harnessed for cancer therapy. And then I'll describe an aspect of that, namely antibodies as treatments for cancer. And secondly, I'll describe cancer gene therapy, which is really at the frontiers. First, the immune system. What does the immune system do? The immune system has two roles. The immune system's job in your body is to recognize foreign substances that are normally not part of your body, and then it mobilizes cells and molecules in the immune system to reject that foreign substance. So really there are three steps in these two functions. Number one, recognition. Second, mobilization and rejection. The foreign substance is called an antigen. Anta means antibody, and gen means generate. Antigens are not necessarily a large substance, but it's a group of atoms linked together in a specific way. You know, H2O, water, always has the same shape. The atoms are put together in a unique shape. Well, it's not an antigen because we have a lot of water in our body. But atoms on, for example, a toxin from a bacterium, like a botulinum toxin, has a unique shape in some of those atoms that we don't have in our body, only the bacterium has it, so the immune system will recognize that. What happens? The antigen provokes a response by the immune system. There are two branches to the immune system. One's called the cellular immune system, and the other is humoral immune system. Let's look at this graphic in a little more detail to describe this. First of all, here is the foreign body, or it's got the antigen in it. And the most important aspect to get the immune system provoked is the following. The immune system is present in lymph nodes and in cells in the blood, etc., sometimes in the skin. But it has to be, the body has to know that the antigen is there. And that is done by display. There are cells in the body whose job it is to put a flag of an antigen on their surface. If there's an antigen present, they display this foreign substance on their surface and they say, honey, I'm home, I'm here. Come get me. Come mount an immune response against this foreign substance. So it takes the foreign substance and it puts it on the surface. Now remember Alan. Alan's immune cells were harvested and this stuff was put in, right? So they were stocked with this immune stimulant, and that was essentially a recognition phenomenon. The recognition event is recognized by a cell called the T cell. It's a helper cell. And the T cell recognizes that foreign substance and sets off a specific response to that foreign substance. First, there are cells in the humoral system, or the cells in the blood serum itself, that make antibodies against the substance if it's hanging out in the blood. And second, there are cells in the cellular response, called T cells, that essentially kill any cell that has this substance on it. So you can take the substance itself and get rid of it using the B cells or the antibodies, and you can actually kill the cells using the T cells. Here is the phenomenal thing about this system. There are tens and hundreds of millions of different combinations of atoms that are going to be foreign to your body. And your system might be different than mine even. So your atoms might be, the atoms that you regard as foreign might be different to the ones that I regard as foreign. But hundreds of millions of combinations out there. Chemicals that we haven't even made yet that the body can react to. Now, we have specific cellular and humoral responses. That is, cells in the cellular system that can find out a cell that has this grouping of atoms, this specific and unique, one in hundreds of millions grouping of atoms. We have antibodies made that'll bind highly specifically, like a lock and key, to any specific grouping of atoms. Let me give you an example. You have antibodies made in small amounts and cells, T cells, made in small amounts that recognize HIV, the virus that causes AIDS. You have a small number of those soldiers. They're constantly being made in small numbers. And if the draft happens, in other words, if you are infected with HIV, all of a sudden that army gets expanded. It gets expanded when this recognition event happens. When the recognition happens of a cell displaying HIV on its surface, then all of a sudden those cells will be mobilized. So it's an amazing molecular system, details of which are another course's worth, really, to figure out how the body can make so many different types of antibodies and cells to recognize foreign substances. So after mobilizing, the recognizing cells send their signals to their siblings to divide these small number of armies, to divide rapidly now. They form a clone of identical cells, and the clones march and kill cells that contain the foreign substance or the antigen in the blood. It's a phenomenal system. In cancer, very often the body makes new proteins. There's mutations. It makes a brand new fusion protein, like the basketball player in the first lecture, making a brand new protein in his white blood cells, and so the immune system might recognize that protein. However, remember, that protein is hiding out inside a cell. So unless that cell breaks open somehow, it's going to be hard to display it. So if tumors make new proteins, the immune system can hunt them up. So here it is. It's hunting them up, it's recognizing them, and then we'll kill them. And there's a photograph there of a false color photograph of the immune system killing them. So the immune system cells have to be stimulated somehow because sometimes tumors evade the surveillance of the immune system. They might have the antigen, as I mentioned, hiding out inside the cell. And unfortunately, the immune system can't look inside the cell and say, oh, there's one. No, the cell membrane blocks that. So you need a mechanism to stimulate somehow the immune system. Well, our opening story described, right, Alan's own presenting cells. You'd add an antigen to it. Here's another way to do it. Here's a protein called MUC1. It's expressed on the outside of many different types of tumors, lung cancer, breast cancer, colorectal cancer, ovarian cancer. Here is the MUC1 protein. So we can isolate that MUC1 protein, encapsulate it in some way, inject it into the body, and hope that the immune system will say, oh, foreign, and then essentially mount an immune response against it. So that's essentially a vaccination phenomenon. It's a vaccine because you're adding the antigen and you're provoking an immune response against the antigen. Here's the result of MUC1 vaccination, and it's quite a spectacular result. This is a Kaplan-Meier curve of survival against time in months, and the survival benefit is just terrific. And this is lung cancer. So its survival is normally 24 months, and the survival is much, much longer now in the vaccine. Obviously, that's under clinical trials. Another way to stimulate the immune system is to stop it from getting tired, okay, essentially. When you react to something that enters your body and you mobilize the immune system and you kill whatever's there, the immune system then shuts down. Why should you have the army keep going? Why should you have complete mobilization of the army if you don't need it? So you demobilize the army. Essentially tell the soldiers all to go home. The protein that does this on T cells, the cells of the immune system that are the keys to the immune response, is a protein whose name is called CTLA4. And what this does is acts as brakes in the immune system, and when the system is no longer needed, it essentially allows those cells to go into programmed cell death. A scientist, James Allison, at Memorial Sloan Kettering Cancer Center in New York, has shown that if you block this CTLA4, this molecule that essentially tells the immune system, well, stop now, it's over, you'll get a stronger response. Of course you will. So a drug has been developed by a small company. The drug is called Yervoy, and the drug binds up CTLA4, stops it, essentially stops the CTLA4 from acting, and the CTLA4 is no longer brakes on the immune system, and as a result, the immune system keeps fighting the cancer. The results of 10,000 patients being treated, some of them are pretty spectacular. It's no wonder that the small company that developed the CTLA4 anti-therapy, the anti-CTLA4 therapy, was bought for $2.5 billion by a major drug company. In addition to looking at the cells of the immune system, we can harness these antibodies. You remember antibodies have specific three-dimensional structures, and they will bind to any of the antigen that's in the blood system. Early in the course, I described some targeted therapies used for breast cancer against a cell surface protein. And let me just go over that a little bit now in the context of immune therapy. There's a growth factor called HER2. And its receptor is on the cell surface. And so if we could bind a drug to that will unlock, right, stick into the lock, essentially we would block the growth factor from binding. The drug that we are going to use is an antibody. This is a graphic of an antibody. Here are all the atoms of the antibody. And here is the antibody itself. It looks like kind of a Y. And here's the antigen. There are two places in the antibody where this specific interaction binds. In 30% of breast cancer patients, a growth factor receptor called HER2 is overexpressed. and that contributes to the highly aggressive nature of the tumor in those patients. A physician at UCLA, University of California, Los Angeles, Dennis Slayman, did painstaking analysis over years, testing out antibodies against HER2 for therapy, first in animals and cells and then in patients. And finally came up with an antibody, which is called Herceptin, that worked. Essentially, it binds to the HER2 cell surface and essentially blocks the growth factor from binding there. So here's the situation. Normal breast cells make a small number of these receptors. Breast cancer cells, through gene amplification, make a large number. And the antibody then binds up the receptors, ties them up, so the growth factor can't bind, so the cell can't get stimulated. Here is the initial result, and it was absolutely spectacular. It's a Kaplan-Meier curve showing that over a 5-year period, 85% of the patients will survive, whereas 67% would before. It's a 20% improvement in survival, and this is a lot of people because it's breast cancer therapy. There are now antibodies for use against many cell targets. And so there are antibodies used against many cell surface molecules in many different types of cancer. Here are some of them whose names you will see. Herceptin, of course, we have just learned about. It's the HER2 receptor in breast cancer. Avastin, we have already discussed in the course. That came from Judah Folkman's work on angiogenesis. It binds the signaling molecule, VEGF, that is needed for essentially growing up angiogenesis. There is a molecule called Erbitux that binds another receptor that's in skin and colorectal cancer. Yervoy, we just talked about, CTLA4, that's an antibody as well. Rituximab, Rituxan, blocks a receptor in lymphoma cells that stimulates them to divide, and so it's used now in treatment of lymphoma. All these drugs, with the exception of Yervoy, are widely used in a lot of different tumors. Now, in addition to antibodies just binding to the cell and stopping the normal stimulant from binding or acting in some other way, you could couple the antibody just to make sure we're going to kill the cell. Let's couple it to something poisonous. So people are doing radioimmunotherapy. They're taking the antibody and they're adding to it a radioactive substance. So when the antibody binds to the receptor on the cell surface, it'll not only stop the normal stimulant from binding, it'll fry the cell itself. So you can couple it to radioactive iodine, etc. So here is an example of radioimmunotherapy of lymphoma. Here is the antibody binding to the cell with this radioactive isotope. In this case, it's radioactive iodine. Here's a Kaplan-Meier survival curve showing, and this is on a year's basis, showing if you add chemotherapy plus radioimmunotherapy, you're getting 87% survival versus 64% of conventional chemotherapy alone. So again, very spectacular results using immunotherapy. I mentioned another way, gene therapy. There are several strategies for doing gene therapy. Well, we got to start by looking at what genes we're talking about. Back to oncogenes, the gas pedals stimulate cells to divide, and tumor suppressor genes being mutated, so we cut off the brakes. Okay? And you'll recall, we know biographies of oncogenes and tumor suppressor genes for a lot of different tumors. Here it is for colorectal cancer, and as I said, for other cancers, we know a lot of these specific gene changes of oncogenes and tumor suppressor genes. So here's the idea. If you've got a tumor suppressor gene that's bad, it's not working, the brakes are mutated, let's add the good one. Okay, let's put in the good one. Now, we can't do gene surgery. We can't go into every tumor cell and say, there's an A there, put a T in instead. Because there's billions of cells. But instead, we're just going to put in, somehow we'll just insert the normal good gene. Okay, that'll be gene therapy. Otherwise, we could do some gene therapy to block oncogenes. So in that case, we're going to block the oncogene from expressing itself. And we'll look at examples of that. Most gene therapy, actually, is done on cancer. It's done on somatic cells. Some gene therapy is being done on inherited diseases of other types, but most of it is done on cancers. 3 types of gene therapy. Mutation compensation. You're adding you're overcoming mutation by adding the good gene. RNA interference. We're going to stop gene expression. That's going to be useful for oncogenes. And finally, something really amazing, which are tumor-specific viruses. Gene therapy of cancer at this time is done usually on very sick patients who have failed other therapy. It's typically observational. These are not phase 1 trials even. They're just observational. Let's just see if this does any good, because this is really at the edge. You don't go in to see a doctor and say, I want some of that gene therapy that's supposed to be really good. We're not nearly at that stage. But here are the methods. So first, mutation compensation. Add the good gene. How are you going to do that? We're going to need to put the gene in the cell in a rather clever way. And the clever way that we do it is by a virus. So we'll take a virus that normally infects the cell, and we'll put in a good gene. And in this case, we're going to put in the gene for P53. P53 gene is mutated in many types of tumors. For example, here, I'm going to talk about lung cancer. And P53, you recall, is a tumor suppressor. It's involved in stabilizing the genome. So if it's mutated, we're in big trouble as a tumor. So we're going to put in a good P53 gene. And we're going to get in by using this vector. So all we do, it sounds trivial, but it's not. We take a virus that normally infects lung cells. We disable it. We take its DNA out, and we take out the genes that are going to be involved in causing bad things, causing the virus to reproduce inside the cell. So now the virus is only going to get in and put in whatever it's carrying. And instead of those genes, the DNA of the virus that essentially make it reproduce inside the cell, we're going to add a good P53 gene, non-mutated. And indeed, the way this was done was to essentially just spray it by a nasal tube onto the tumor in the lung. The result is shown here. Here's just one result of a CT scan. Pre-treatment, you can see a very large tumor, and post-treatment, the tumor has shrunk. They showed the normal P53 gene was there and was being expressed. This is at the very edge of cancer research, the very edge of trying treatments. The patient shown here died because the patient was dying at the time. But the patients will consent to this because of the small chance that it might do some good, but also it's going to do some good for people later on. So participation in clinical trials is very important, not just for dying patients, but for any patients with cancer sometime in many instances. Okay, what do we do about oncogenes? We want to tamp it down. And the way we do that is what is called RNA interference. In the 1980s to the 1990s, scientists, biologists literally using first flowers and then worms, two different organisms, found a brand new and undiscovered way that the body turns off gene expression. What they do is, the body does is, it takes small RNAs. The typical messenger RNA might be 1,000 or 2,000 of these building blocks long. Messenger RNA, remember, is that copy of the blueprint to make a protein. And the body, to turn off them, it makes the messenger RNA, but it says, I don't want to use it. I don't want to use it. The RNA's in the cell. I don't want to use it anymore. It makes a small RNA, and a lot of the genome is making these RNAs, it turns out. Makes a small RNA, and the RNA then binds to, by base pairing, this target messenger RNA, and essentially the messenger RNA doesn't work. Now, when people discovered this in the plants and the model system and worms and in animals and in human cells, it was very exciting because right away we see the application. The application is, of course, we could turn off oncogenes. The people who discovered this, of course, won the Nobel Prize. A small company that was working on this was bought for $1 billion. Billions of dollars are now being invested in this potential of this therapy because of wide applications in medicine, not just cancer. So here's the idea. It's interfering with RNA. You block RNA expression, and essentially the RNA gets destroyed. It works in mice. Now it's in human clinical trials. It's going to be difficult, but it's beginning to work. Not in humans yet. Third, tumor-specific viruses, the amazing therapy. So here is a virus that infects a cell. Cancer researchers have made viruses that will only reproduce and make new viruses and destroy the cell in the process in tumor cells. It's an amazing thing, and there are many, many strategies to do this now. So here is a virus called an oncolytic virus. It infects a tumor cell. The virus replicates, more virus, tumor dead. When it infects a normal cell, it doesn't replicate and the cell is undamaged. That's a great therapeutic ratio, isn't it? Right? And here is an oncolytic virus treatment. And you see here is a tumor and colorectal cancer, and after treatment with the virus, the tumor shrinks. Again, the very edge. This is the very edge of cancer research. Lots of animal work is being done here. The excitement is you're targeting a tumor only, and you're not targeting normal cells. So this is really the leading edge of cancer therapy. So in this lecture, I've described harnessing the immune system. I've described gene therapy at the edge of cancer. But we're still not doing well in cancer treatment. 500,000 people are dying in the US every year from cancer. 7 million die worldwide. Knowledge and science is resulting in new therapies and optimism. But while these new therapies are developed, we can do things to detect cancer early enough for current treatments to be effective and even prevented by avoiding some certain activities. In the next 2 lectures, I'm going to turn back from looking inside cancer cells to where we started in the course, at the big picture level in screening and prevention. Thank you.